Kwewacha, hello everyone. So even if we're remote, I'd like to take a moment to thank you for welcoming us on your respective lands. Thank you for being with us today to talk about surveys. I'm Raphaël Pichet. I'm project lead for conservation at FNQL SDI. We're going to hear Rebecca Lacerte from Université du Québec à Rimouski. She'll tell us about how to do a fauna survey and what are a few common methods to do so. So Rebecca is a PhD candidate with Professor Martin-Hugues Saint-Laurent at UQAR. Her thesis is about weather conditions and its impacts on productivity of food resources, ecology, and individual performance of animals. She studied with the Esipit Nation in collaboration with them about closing forest roads and the benefits for the woodland caribou. She's also teaching surveys at UQAR. This morning, she offers a special seminar about large fauna surveying. Rebecca, please, you have the floor. Thank you. So, hi everyone. So, first, let me introduce myself very quickly. Or actually, I was actually already introduced. So, let me just tell you that the content here is part of the class that I teach along with Martin-Hugues Saint-Laurent. So, he's a co author for the presentation today, even though he's not attending. Okay, so why surveys? Well, first, we want to know what's going on the land. We want to know, for instance, what's going on with a given population. These surveys are a part of a larger domain, which is ecology. Ecology is a science that requires real-world data, which is why we collect data in vivo, outside, out in nature, in order to test hypotheses. The hypotheses are basically the questions that we ask about the land. We can carry out surveys on any organism, from fish to amphibians to birds to plants to insects. As if we wanted to, say, have a survey on the lynx, we could definitely broaden that to include rodents as well, since they interact. What can influence the lynx, is everything on the screen here. So everything is interconnected. So if we want to carry out a survey on the lynx, ideally, we need to consider all the applicable parameters. These phenomena are complex and these interactions are multi-layered. So it's important to know what's going on, but we can't measure everything, right? Because this is a lot of time and money. And we can, everything can go down to the quality of the soil, for instance, and that will have an impact on everything else. So we need to narrow down which parameters are worth our attention. Today, we're really going to focus on large animal survey. So mostly wolves, caribou, this kind of animals. And I'm going to give you a few examples of questions that we may want to ask ourselves. So we may wonder how many individuals on the land of a given species. We could wonder what they eat. So what do they eat on this specific piece of land? We could also want to know what species are found in a given region or area. We can also wonder if some species are where they're coming from and where they're going. We can wonder when they migrate and why they do so. We could ask which species is predating on others. We could ask if a given population is more abundant or less abundant than before. We could also ask a question about how many individuals we can harvest so that the population can stay at a healthy level. So once we have these questions in mind, there are steps to follow. So first, we need to narrow down what question we want to focus on. The second step, we need to pick a method, a survey method to answer this question. Then we create a protocol. Then we collect data. Following that, we analyze the data. And finally, we use the results. There are many ways to do that. And sometimes these results lead us to additional questions, and we go and start over again with this cycle. So here in this process, Indigenous knowledge can be beneficial with these steps. I'll get back to that in a little bit in my presentation. So first step here, state the question. This is the very first step. Questions can come from communities. So why are there less walls or more walls in a given area than before, for instance? Of course, Indigenous knowledge can bring a lot to these questions. So it's really focusing on the issue or the question at hand. And this is typically coming from the people who use the land and who live on the land. Let's use an example. If we have a given issue here. So the coyote, for instance, the issue is we don't know how many coyotes are living on our territory. So we want to know how abundant coyotes are. If we pick this objective, then we need to think through how we can answer this question. This is a simple example here, to determine the count of coyotes in a given area. This is fairly easy to do, but not all questions can be answered. Please bear that in mind. So some objectives or some questions will simply be impossible to answer. This is important to know before we launch into the survey. We need to ask, is my question answerable? Do we have methods that can help me answer the question? Objectives need to be very clear. So determining how abundant a species is basic, and we want to narrow it down a little bit. So where should the survey be held? What are the borders of that area? Are we surveying just one area or several areas? We could also want what degree of accuracy we're looking for. Do we want a very specific number of individuals or an approximate number of how abundant the species is? Or do we want to compare how abundant the species is in several areas? So this is important to know before we launch into the survey because this influences the type of method we're going to use. We could also narrow down the when. Do we want to know how abundant the species is on a yearly basis, or do we want to do that in a specific season? That could change, of course, and ebb and flow with seasons. So we can get a longer list of questions, but the clearer the objectives and the questions, the better you can choose the right method. So picking the method, the survey method here, this depends again on several questions related to the objectives as we just set out. So the where, the when, the species, the what. So these are all questions that can help us to narrow things down. We can wonder here what the best method is. And the last question is by whom? Who is going to carry out the survey? So we can wonder here what methods we're using. So for instance, for estimating how abundant a species is, there are three main questions. So are we able to observe all individuals? Second question, if not all individuals can be observed, what can we do? And then the last one, only signs of presence are being observed. So this could be based on the sound or on footprints, for instance. So if we look at all individuals, then we need to have a full count of a given area. This helps to have a very specific number, but of course it requires a lot of time and money. We can also have partial sampling on parts of the area. So I won't go into the whole details here, but we can have random sampling, stratified sampling, systematic sampling. If we're using just signs of presence, like feces or noise, then this is what we use on the field. Now, if we have the scenario where we're not observing all individuals, then we can simply count the number of individuals knowing that we won't see all of them. Again, there's several ways to proceed with this count. I won't go through the details for all of them, but do know that there are several. We can also capture individuals on the field, which helps us to carry out analyses to determine the full count for a given species. With captures, we can have removal. This removal can be selective. For instance, only males. This can be done with selective hunting, for instance. So this can be a method that will work. We can also have non-selective removal. It doesn't have to be specifically males or females, for instance. We can also do SCMR. So this means we're capturing individuals. We earmark them with tags, for instance, and then we release them and capture them again later on. So this isn't all. This is only a few methods for looking at how abundant a species is in a given areas, but there are several methods to answer various questions. So we're not just looking at how abundant a species is. We could focus on their food or behavior. So many, many methods exist. This is why the objective needs to be very, very clear. This will help us really to pick the right method for our needs. If we want to determine how abundant coyotes are, we can pick the method where not all individuals are observed, and we could use the capturing method or the count method. So several methods can be used to answer the same question. This can depend on our sub goals, but also how much time we have and how many resources we have to invest in this survey. Creating a protocol. Why would we want to do that? Well, we want to know specifically what steps and measures we need to use on the field. We can't just be on the field and not really know what to do, right? So we need to have a step-by-step process. We want to make sure that the measurements meet our needs. If we are on the field and we're not well prepared, we can measure things that in the end won't really help us answer the question at hand. So again, not everything that is measurable is worth measuring. So we really need to know, will this measurement help me answer a question? If the answer is yes, then we put it in the protocol. But if not, we let it aside. Is the data relevant for our analyses? This measurement, will it be accessible, so to speak? Some data can be interesting, but we may or may not be able to carry out analyses and further steps downhill. So it wouldn't be useful to use these. We can also wonder, will these results be usable? So the outcomes related to a given variable, will they be usable down the line? We need to answer these questions. Also, we want to create a protocol to reduce the number of questions out on the field. If a protocol is well written and clear, we can go out on the field and know exactly what to do and just collect data without asking any questions. So this will be very efficient. We know what we need to do, and it's quicker also, of course. And this means that it is cheaper also because people know what to do. It also needs to be very clear and specific to be used again for other teams or, say, other communities, for instance, so that we can carry out surveys on various territories and also carry it out again year after year, for instance. So then we can have a time-based view. Sometimes, of course, the teams on the field will change. So we want to have the same method every time. We want to make sure that data represents reality. We want to have a protocol that allows us to have data that really represent what is going on the land. Over here, here's an example of what this means for the data to represent reality faithfully. We have a given area here. You have the green areas here that represent various types of trees. We have here a few samples that represent the abundance of a given species. So in yellow, we can say the abundance is lower, and in red, there's a stronger abundance. if we look at this grid, if we pick the white squares here, so really with hardwood species, we see that the abundance is high. So picking just the hardwood trees will give us poor results, because if we carry out our analyses afterwards, it means that in the territory, on the land, the abundance will always look like the hardwood areas. So we're overestimating the numbers here. So here the text says exactly what I just said. What I'm trying to say is, if we're not considering the variations on the land of given species, then we're going to overestimate or underestimate how abundant a species is. Okay, so how should we create a protocol? Again, there's dozens of methods. It can get confusing at some point. But when we, again, have a clear objective, it's extremely helpful. So there's people who came before us and they've probably done something like we want to do. We can look at what has been done before. We can look at reports and studies to help guide our choice. When we have a literature review and have some ideas, so we can ask, if people were trying to answer the same question as we are trying to answer, will the method meet our objectives? If no, then we can just remove these pieces of literature. We also ask, will the method be applicable to our species and our land? Some methods will be used, for instance, on plain areas that will not work in forest areas. So we remove those also. And of course, do we have the budget, the time, the staff to use this method? Some methods require too much resources, and then we can remove those also. So then we're left with just a few articles, a few papers to help influence the choice of protocol. Among those, some will say in their papers, we use this method, but we had some issues, and they can list out the issues that were observed on the field or when they wanted to carry out their analysis. So we can use these and also the specific methods in order to create our own protocol. So we can see here in a detailed manner what was done. And we can take several perspectives to create our protocol. We, of course, want to avoid running into the same problems that have been observed before. What are the advantages, the benefits of a clear, specific protocol that meets our objectives? Well, again, the collecting of data is efficient. It reduces costs. So the more effective we are, typically, the lesser resources are needed. And we can also make sure that we're not doing anything that is unnecessary. So we're not wasting time using measurements that we won't be using down the road. This helps also with comparing results with other surveys on other lands by other entities. For instance, even if the protocol isn't exactly the same, if the protocol is clear, we can compare the results. And also, we can also look at any issue if the analyses are troublesome, if there's a variable we can't quite analyze, then we can go back to the protocol to try to maybe have another measurement, another variable, something to control that aspect.
Now we're looking at data collection. So we had a few questions related to our objectives, and then how did we choose our method at this stage? And then it can be by whom. Data collection doesn't have to be done by ourselves. It can be by others. We can hire people for data collection. We can do it ourselves as well. So for data collection, once the question has been thrown out there, that the objectives are clear, the protocol is clear, we go out in the field for data collection. And this is the part that is the easiest, if you will, for an inventory, because once we're there, all of the questions have been answered. all of the problems were solved for the most part, and we're just out there on the field and collecting information for our inventory. Those that will gather data, it's not always those that will have come up with the protocol. Those that collect data must be aware of the objectives of the inventory. Why? Because if the problem arises on the field staff that is collecting data must know the survey objectives and the analyses that will be done after that, so that they are able to modify the protocol without adversely affecting the analysis and therefore still meet the objectives. So this can happen in spite of all the questions we had, the problems we might have resolved or think we resolved during the creation of the protocol. It's possible that when we get out there on the field, there might be an issue that we hadn't thought of. We must therefore change our protocol. So if those that collect data are aware of this issue and they take the objectives, this is why, so they need to know why the protocol was created this way. And so then the protocol will be able to be changed so that the measures out on the field can still meet the objectives, the basic ones, and will still be able to find answers to the question that we had to start with. Analysis. Why do we have analyses? First of all, if we have a question for an issue concerning abundance, unless we do a complete census, this is when we count all of the individuals on a territory. We include 100% of the individuals. If we don't, we must have analyses to know what the actual abundance is. So we need to know what the inventory is. For example, if we count 100 moose, it doesn't mean there are 100 out there on the territory because we saw 100. We need to do an analysis. We need to include in our analysis the fact that we don't see all individuals and also that we don't sample the whole territory. If we use comparison objectives, for example, here we want to compare between the territories, analyses have to be done to know where those differences are. Are there real differences? And why? What treatments are different than the others? So we need analysis to know where the differences are between the territories are. For diet objectives, analyses are necessary to identify the foods mainly concerned, and analyses might be done in the laboratory, in the lab. To this effect, the type of analysis is directly linked to the selected method. When we choose a method, there's almost always an analysis that is directly associated to that method. For example, if we have a type of inventory, an analysis will be directly related to that type of inventory. Oftentimes, data analysis, there aren't that many questions to be asked about the type of analysis. It's the method that we will be looking at. When our objectives are well defined, we can easily get those analyses done. And also when there's a clear and precise protocol and there's a rigorous data collection, it allows us to come up with really good results. So the expression here, garbage in, garbage out, means that when we do undergo analysis, we're able to include everything in that analysis. These remain numbers. We can put anything in there and we'll come up with results. Are the results good? Will depend on how we considered those data. So if the data is good, our results will be good. And inversely, if they are not good data, our results won't be good. So we need to have rigorous data collection on the field so that we can have good data and come up with good results. We must always remain skeptical and critical of results. Analyses remain statistics, and statistics aren't always, and environmental issues don't always follow statistics. When we create models and do analyses, we'll come up with a result, but we need to remain skeptical and critical because we're still dealing with statistics. We need to keep in our ecologist mind that we need to consider whether the results make sense or not. For data analysis, we could also ask by whom. We don't have to do those analyses ourselves. We can take the data and hire somebody and give the company, pass the data onto a company so that they can analyze it. Sometimes there are complex data that we might not have this competency to analyze. we can give our data to someone else so that they do that. So data analysis can easily be done in partnership. For example, my master's project, I did this in partnership with the Innu of Isipit Council Nation. They did the field work for the inventory. They asked the question, they chose the method, they created the protocol, and they came up with inventories. And once they had their data, they provided us with those at the Université du Québec in Rimouski for master students, myself. This is a step that's easy. You can easily do inventories and then have partnerships with universities that are specialized in research and analysis of data for fauna data. So you can come up with reliable results, and you don't have to play around with statistics if you don't feel so comfortable with that. You can pass that on to students who will analyze those for you. And then the last step is to use the results. So here you have the different ways of using those results for your inventory. For example, it can help you with management. You can manage animal populations for hunting and trapping. You can identify the lack of knowledge to be able to have a complete picture of the species' health. It can also provide knowledge of a species ecosystem present on the territory. For example, caribou, which is present on a large territory. When we talk about that species, we consider their whole habitat, knowing that they have an important or significant distribution over a large territory. It will also help us to have knowledge of a species ecology present on the territory. It can ensure abundance of species for the future generations. So to monitor, we can see if the population is stable, if it's increasing or decreasing, and allow us to continue to harvest through the years if we can have a stability in that population. And also self-determination and resource management of territories by the nations. So we're done with our introduction of inventory, so I can answer your questions, and then we'll go into the presentation of more specific inventories. So we have what Rebecca presented to us, how to establish our question, our protocol for establishing an inventory. So you can raise your hand in the reaction button at the bottom of your screen, or if you prefer, you can also write those in the chat. If you prefer me to ask those to Rebecca, you can ask your questions in English or in French, because we have simultaneous translation. There's no problem there. So there's a question from Pierre-Luc Bastien from Kirigan-Zibi, who said, what's data sharing like between communities and access to these data? Who has access to that? I don't know. Between communities, I'm not sure how that takes place. I'm at the university here, so between communities, I'm not sure. But you, with the Isipit community for your master's degree, how did that happen? Well, if I take my example, it's not always the case, but the community approached me, came to see Martin Serie, who works with large fauna, and they communicate with him for his project. And then we had meetings, everyone together, so that they can tell us what their objectives were, what their question was, what they wanted to know. And then they provided us with their data. And with those questions that they provided, I analyzed the data in light of those questions. And from time to time, we would have meetings to see where I was at in my analysis. What initial results did I come up with? And I provided them with reports that, in light of those results, we had meetings for these results. And I also gave conferences with regards to my findings, and articles were published as well. My master's thesis was also published as well. And if I can add, for the data, so the community went and got those data on the field. The data belongs to them. Yes, that's right. So, yes, because Pierre-Luc specifies that the question of confidentiality or privacy as data is often an impediment for collaboration with universities. I worked on this type of project. There's always a confidentiality agreement, an ownership agreement that's signed between the community and the university so that we know who owns that data. A very good point. Thank you, Pierre-Luc. And any other questions? I have a question, Rebecca. Will we see later on what the best methods are for data collection? What application, what program, will we go that far? I will show types of inventories. I won't go through all of them. And to see which one is better than the other, it depends on the question. So there's not one inventory that's better than another to start with. It depends on the questions you have to start with, and then you choose your method. Sometimes there isn't a best method. There are 3 or even 5 methods that could reach those objectives and answer those questions. I will present how the inventories are done on the field, but I won't go into analysis after that because that's more calculation. And it becomes a bit complicated to present that. It is not necessarily. We don't remember all that after a conference. But the type of application that you use for data collection on the field, what kind of software would be more practical out in the field? Because I've worked with iPads, but is there some software that is that is easier than others? I wasn't going to talk about that, but I could. What's easiest is to use paper. If it rains, it's ideal, right? If we're in a helicopter, it could be with an iPad, but after that, it's to enter the data in an Excel file. If the data is in an Excel file, we can do all the analysis we want with the data. It's the best way for data conservation. Maybe I can add too, when Rebecca talks about paper, there's special paper right in the rain. For those of you who don't know, you can use it even if it rains, right in the rain paper. I don't think there are any other questions. You can continue, Rebecca. Okay, great. So I'll go back to speaking slowly. So the types of inventories that we'll look at today, we'll look at aerial, DNA samples, and cameras. So I'll start with the aerial surveys. I think we'll take a break after the aerial surveys. Yes, perfect. So I'll start with this before the break. So here we go. So aerial surveys, first of all, when we look at the graphic that I showed you earlier, most of the aerial surveys will be in a category where we don't observe all of the individuals. And we'll do a certain count. And we can do this according to 3 different methods. It can be double count by marked individuals, and it can also be a linear transect. So what is an aerial survey? It's counting individuals from the air. And this meets which objective? Mainly, it's to answer questions linked to the abundance of the species. We want to know what the number of individuals is for that territory. It can help to identify the distribution of the species. And how is this done? Usually, for these surveys, the territory is flown over by helicopter or by other aircraft, and we will count the individuals that we see when we do these inventories. Usually, the aerial inventories, it will be done in wintertime, but it can be done in any season. So aerial surveys, not all individuals are observed usually. So when doing the survey, we must also determine the detectability. That is our capacity to observe those individuals. Detectability is how many individuals can we actually observe? What is our capacity of observation? We know when we do an aerial survey that we won't see all of the individuals on the territory. And the detectability, for example, do we see only 80% of the individuals or 20% of the individuals? The level of detectability, it doesn't matter if we only see 20% or 80%. What's important is to know what that percentage is, to know how many we are observing, what that ratio is. No matter what level of detectability, through the analysis, we are able to know what our abundance is. To determine this detectability, there are three methods. Subsampling with marked individuals. So marked individuals will allow us to identify how many individuals we're able to observe from the air. We can do sampling with more than one observer. A second observer will allow us to see how many individuals that the first observer might have missed, and this will help us for detectability. And finally, we can count on transects. Detectability will depend on transects, and I'll explain a bit later how we can do that with this method. If we look at subsampling with marked individuals, obviously, to do this type of aerial inventory, we need to have marked individuals on the territory, and we need to know how many of them are on the territory. Marked individuals can be colored collars or tags, telemetric collars. This can be done many ways. For example, here we have a territory and we have an aerial survey in a certain sector. And so we'll do a survey in this sector to count the individuals. And we have telemetric collars for some individuals here. So we will see how many we observe. This allows us, first of all, to have our inventory with our number of individuals. And for example, in our aerial survey, we counted 106 deer in our sampling plot, and of 10 marked deer, 8 were seen. So we know that we have a detectability of 80%. And so we detected 8 out of 10. So with that percentage, we're able to calculate the abundance of the individuals with our aerial survey when we counted the deer that were in that plot. Now, if we change types of aerial survey and we go into sampling with more than one independent observer, that means that the observers don't talk to each other. It allows us to estimate detectability without marked individuals. If we have no marked individuals on a territory, we can do an aerial survey with 2 observers in the helicopter, for example, allowing us to see the number of individuals we see. So the first method, we talked about marking with a tag. The observers will identify the individuals in a geographical position, a GPS, and this will allow us to detect those that are marked. Each observer will do their own inventory. They will identify the individuals on the territory, their GPS location for the individuals on this territory, with 2 different inventories by 2 different people, but in the same helicopter. We're able to compare these 2 inventories and see which individuals are missing by one or the other of the observers. And that way we can account for the percentage of detectability and then establish the inventory. So 2 observers placed on the left side of a helicopter, 1 in front and 1 in the back. And in the helicopter, we can have a window that will allow us to see on the ground, and we will count the individuals in that window. That's how that works. A second type of aerial survey, sampling with more than one independent observer. So it's the same principle as the previous inventory, except that the observers will do the inventory together. They will be speaking together. So how that works, there's a primary observer. That person will count all the individuals they'll see. And then the secondary observer, their role is to identify the individuals that the first observer missed. With those missed individuals, this is where we'll be able to identify detectability. For aerial surveys, we can also count on transects. This counting on transects, it's just a straight line that we will oversee on a territory. And this line can be done either by car, walking, or it can be an aerial view as well. If we take an example here, the green dot here is the position of the observer along the transect, and the transect is the blue line. That's the walking line, the straight line. And the principle here is that the observers will walk along the transect, and when they see an individual, they will note a certain measure that will allow us to come up with a certain abundance. If we see an individual far off, the observer will note, first of all, the distance estimated or measured between the observer and the individual. We will also want to use the angle, and these two measures will allow us to calculate the perpendicular distance between the transect and the individual. And this is what will allow us to evaluate detectability. We are walking on this transect. The chances that we observe an individual directly on the transect are quite high because the individual is close to where we are. And the chances that we observe an individual that is far from the transect, that individual is far away. We could miss it. It can be hidden behind a tree, for example. The further away this individual is, the less chances we have of observing it. So with that distance, we are able to see what the detectability is. And so the text here indicates exactly what I just said about the distance for estimated or measured detectability. So that's it for aerial observations. Do you have any questions with regards to this inventory? I can answer those, and then we'll have a break. This is Casimir Boivin from Manawan. I just wanted to know, the observers don't communicate with each other. Why can't they communicate? Is it to have a better result? Yes, exactly. The method for by not communicating, we don't want any bias between the two, because to have two different inventories in a helicopter is to see to what extent we miss certain individuals. If they communicate together, they could say, oh, don't forget such and such an individual. The inventories would be biased between them because it's not only the individuals observed by one observer, it would be the individuals observed by two observers. The goal of not communicating together is to have two completely different inventories to be able to say, when we have that inventory, here we have so many individuals, so we'll have a certain number.
Okay, thank you. Yes, Flavie. Thank you. Hi, Rebecca. I have a question about detectability, especially on the first estimates. You were saying we compare the number of animals that we observed. Can there be bias with regards to the fact that some of these animals have already been captured and maybe they're the ones that are less careful or easier to spot than others that are better at hiding? Yes, there can be a bias related to that. These are typically included in our analyses with capture, tagging, and recapture. If we do aerial surveys, we don't do this capture and recapture method. So this reduces this bias. So if there isn't a concept of recapturing, then that's one thing. If we do have recapture, then it needs to be included in the model that previously tagged animals can be recaptured. It goes both ways. So individuals, depending on the capture method, sometimes we can use things to attract them, a lure. So some individuals will be scared to go back because of the stress related to being manipulated, but others will go even more because they're seeing the reward with the lure, and they don't really mind the manipulation aspect. So the benefit of the food is greater to them than the discomfort of being manipulated. So this can go both ways, and there's ways to control for that. And second question, when we can't estimate detectability with the method, if we have two observers in the same device, if the helicopter doesn't go over zones where there are animals, how can we calculate and compensate for that? So this is really with regards to the number of individuals that we can see in a lot. So if in a lot there's supposed to be 100 individuals, but we only see 70 with a 70% level detectability, then this isn't about any parcels or loss that we aren't seeing. It's really detectability is just, can we see them? Can we spot them when we fly over them? And we really need to have plots for sampling, and this is taking into account that some plots can contain individuals, and therefore we really need to run some averages. So we have several plots, some that will contain no individuals, some that will contain some individuals, and some that will contain more than the average plot. And so with our analyses, we mix them all up to determine the overall abundance. Are there specific methods applicable for a specific species? Because it can, be different from observing bears to caribou, right? Yes, it does depend on the specific species. Some species are easier to spot with an aerial survey with two independent observers. In other cases, it's easier to have a full count. So I don't know the right method for a given species. This can be found in literature, however. But each method, or in other words, each species is related to some methods that will be easier, more applicable to them. Yes, and I believe we have, we have some idea of that in literature. To your knowledge, has this been done with drones versus helicopters? Yes, it can be done with drones and also thermal sensors, so we can see individuals differently. But again, these are methods that are still being tested out. The methods aren't perfect. So for a heat sensor, for instance, we can have service that will lay at one spot for a long time. So the heat will remain even after they're gone for a while. So it's a little bit difficult to know if the individual is actually there or if there's just residual heat. Yes, and let me add to that about drones. From what I heard, people who are starting out with that, there's an issue with the distance from the transmitter. So we can send the drone away only at a given distance from the transmitter. So this does bring limitations. I'm not an expert at all. It's just something I've heard from people who have dabbled with this. Yes, I use drones quite a bit. Recently, there's a peninsula here, and we were surveying the peninsula, and I was doing that with software programs so that the drone can go around by itself. But sometimes there's a connection with a tablet and with a computer, and sometimes we lose a connection. So if there's no Wi-Fi out on the land, sometimes I need to configure things so that the drone will come back, and this adds to the number of flight hours. And if there's any mountains, for instance, then that will also block the signal. Any hills or that kind of thing. Thank you so much, Casimir, for this very relevant addition. Thank you for sharing about your work with drones. We have a question from Pierre-Luc from Kitigan-ZB. Could we have helicopter surveys for First Nation without the participation of biologists from the department, considering the enormous budget needed for this type of activity? Well, Rebecca, I can answer that actually. The answer is yes. Some First Nations have done this without the contribution of any biologists from the department. We have one nation that will present this tomorrow, their aerial survey of moose. So yes, this requires a very significant budget. There are some grants for that. Do you know that for knowledge acquisition? So if you want to know more about the grants, you can definitely write to us at conservation@iddpnql.ca. So I really encourage you also to be back tomorrow morning to hear about this First Nations experience with that. Oh, I just wanted to add something. Yes, please. With drones, when we talk about drones here, There are various brands, of course, and some may have a radius of X number of kilometers. The one that we have is a larger drone, and I think it can survey 50 kilometers, but we still need to make sure that there's no hill. So if it loses connection, it does keep flying. And there's a return home mode that then is triggered when they lose connection, and then they go back to their launching position. I just wanted to add that. Okay, thank you so much for the details. All right, so now moving on to surveys via DNA sampling and via camera. Starting with DNA sampling. When we carry out DNA-based sampling, this is under the category where all individuals aren't observed. Then we go onto capture, then we go to capture, marking, recapture. Why does this fall under this category? Well, it's because we can tag individuals without necessarily using colors and such. So with a DNA, we're able to know which individuals are captured. If we look at DNA sampling, we can know which individuals were recaptured and which ones were not recaptured. So what exactly is DNA-based surveys? Well, we typically use animal hair to have DNA. We can also use feces or muscle. Now, which objectives fit with this method? Well, typically questions linked to the abundance of a species. If we do capture, marking, or tagging and recapture, we can also study the population diet in this way. So how is it done? We take samples of fur, muscles, blood, or feces. Then we carry out DNA analysis. This is done in a lab. So we simply sample on the field and the rest is sent to a lab to get the DNA from these samples. When we carry out this survey, it's mostly done in the summer and fall, but it could be carried out in any season. So with regards to CMR, capture, tagging, recapture, we're using a software that's called Density. This is the main piece of software that we use for this type of method. Let me show you an example with black bears. So we collected fur samples. The survey was on over 5 weeks. So we're visiting the various plots weekly for 5 weeks. These plots are either 25 or 50 square kilometers in area, and the visit intervals is typically 4 to 15 days. We went weekly. So what we do is we have a bait. This is an example where here you have very clearly marked zones in a square fashion with the trees. Of course, that's a little bit simpler than reality. But we again are choosing plots that are 25 to 50 square kilometers, and we use a piece of thread, two pieces actually, one that is 30 centimeters from the ground and the other one 60 centimeters from the ground. And then bears will walk through the threading to get the bait and leave some fur behind in this way. We're using barbed wire to do this, to catch the hair. So we see the barbed wire here. This helps us to have the size of the plot. So we have this again around the trees and it needs to be very tight. We want to make sure that the barbed wire is really straight and nice and tight. And then bears will simply go through, either going under the barbed wire or in between the two wires. And there you go, you're catching some fur in this way. It just collects a little piece of fur. This is not really invasive at all. So then we pick up the lump of hair, and this is what we send to the lab for analyses. In between each, we're burning the barbed wire Because if we were to leave the hair, this would, of course, carry out biases for further sampling. So we burn the barbed wires to make sure that there's no hair left at all. Then we carry out genetic analyses. We get the DNA at the root of each piece of hair. We need good quality hair to do that. If there's only 2, 3 hairs, then that won't be enough. We need a little bit of a lump with the root of the hair. So we're not sending all the hair to be analyzed. We send only the good quality samples. So this is what the density software looks like. We have the various plots, and for each one, we can look at each individual where they were, and then we can calculate the abundance of the species. Now, if the objective is to study the diet, not abundance, but diet here, what can we do? There's a method that's called stable isotope analysis. Stable isotopes, to make it simple, it's a mix of carbon and nitrogen that we find in all individuals. So we, as humans, we have a specific ratio of carbon to nitrogen depending on what we eat. So everything that we consume modifies this ratio. What this means is that we have a graph like this. So one isotope and then a second one, and each of food represents its own ratio. So each individual has its own overall ratio, but each type of food has its own ratio. So, for instance, plants or vegetation will have a specific isotope profile, and rodents, for instance, will be down here. So this would represent the diet resources on a given area. If we're sampling coyotes, for instance, and we plot these different foods, then we can plot that and we can know the coyotes is in between rodents and vegetation. So this means it's about 50-50 made up of vegetation and 50% rodents. If we take a second example, the black bear is plotted over here, so closer to vegetation than to rodents. So its diet is about 75% vegetation and 25% rodents. This is an example. This doesn't represent the real diets here. It's really just an example with fake data. So if we plot this on a graph, this is a real results here. If we look at the ratios of each individual, we have the coyote, the black bear, caribou, and moose. We can see not only what their diet is for one species, but we can see that there's an overlap between species. So we see that the moose doesn't have any overlap with the coyote, but the black bear, its diet does overlap with coyote and caribou. What this means is that if caribous and bears use the same food resources, this could mean that they're also using the same lands to access the same resources, which can then influence the predation then, because they're finding themselves in the same spot. To look at diet, we can use DNA metabarcoding analysis. So we're using feces in this case. We're extracting all DNA in the sample. We're really getting everything here. So really from all food types, all these different types of DNAs will then be amplified with a machine that's called PCR, a PCR device that simply amplifies each gene. So getting the DNA, sometimes we can just have a little piece of it. But for our analyses, we need to have more of it. So we do amplification to then facilitate further analyses later on. From this, we carry out sequencing of DNA strands. Then we can know what the DNA fits with. We have a reference database called GenBank. So this represents all the possible food sources. So then we can say, okay, here we have DNA that looks like, say, a specific type of tree. So we can know that the bear ate birch. Again, this is an example of an experiment that was carried out in my lab, looking at food competition between the caribou and the moose. So we have a feces sample that were collected at various months from June to September. And you see here specific food resources. So these were all found in caribou feces, and these were found in moose feces. So we see that there's just a small overlap. We can also see over here the difference between high altitude and low altitude. This helps us to see if individuals are using, one type of food in high altitude, and it might be different from low altitude environments. And it can also ebb and flow with seasons. So we can really know a lot about their diet. Okay, so this was all about DNA sampling. If you have any questions, please go ahead. Yes, Eliane.
Still have a question. Your previous slide with the diet, we have the indication on the different types of food, but do we have an idea of the quantity consumed? No, for DNA barcoding, this will be a question of what was consumed. We don't have the quantities, and we'll have a proportion. These are two methods that are complementary. The DNA barcode will pick up everything they ate. It will also include what they accidentally ate. For example, if they want to take a bite of bark of birch and they took something else, it will be considered, and it will be a proportion. What is chosen over others? Are both done at the same time? It depends on the question. Some will want to do DNA metabarcoding because they want to know what was eaten, and others will go with proportions as well if they want to know what has been consumed. And it depends on the specifications, the precisions required. It's not a great answer here, but it will depend on the objective. When you put the barbed wire up, isn't there a risk for the animal to hurt themselves? Or do you use this smaller barbed wire? The barbed wire, the height is maybe 1 centimeter from the bottom of the barbed wire to the top. It's very small. It won't go into their skin. It will just hook up to the hair. I installed some of these. We use these with gloves, but we take these in our hands and it's not that bad. The animal would have to be really stuck to get hurt, and they just go over or under. Any inventory method is never 100% sure. There can always be problems. We've never seen any problems with an animal that would have been hurt. Any blood on the barbed wire, for example. If they go between these, they won't weigh against them. It'll just pick up hairs. It won't stay stuck on the animal. Is there somewhere we can send, if we do data like this, is there somewhere where we can send all the data? Yes, these hair samples are sent to a company that is specialized in DNA sampling. So it's to send all of the envelopes to such and such a company. Many companies can do this. So they will give you a price per sampling, and they'll analyze all of the samples and send the DNA back for each of the samples to those who made that request. Thank you. We have a complimentary question in the chat. Nathan asks, does this methodology have any extra cost to it? Is it accessible? Is it expensive, this inventory? The cost of the barbed wire isn't that huge compared to other inventories. It's just to buy the barbed wire and the bait. It can be sardine cans. For sampling, it depends on the companies. It will also change according to the number of samples. If you send 10 samples in compared to 1,000, of course, the price per sample will change. It depends on the budget, but it's an inventory that's pretty accessible. But for DNA analysis, the cost of a sample can go up pretty quickly depending on the numbers of sampling. So the field that doesn't cost so much, the analysis might cost a bit more. So we recommend getting quotes, but it does change according to the number of samples. It can be a few dollars per sample. It can be much more than that. And are there others for moose, or are there companies that can, for example, if we extract teeth, to know how old the moose is? Or is it the department only that does that? I don't know if there are companies that do that for age through teeth. One possibility would be to work through universities and create a master's project where students could read the age of the teeth. I don't know if any companies do this, but there probably are. Maybe private companies in biology could do that. I would have liked to answer, but I don't know. Pierre-Luc Bastien writes that the equipment required is very expensive. I imagine he was referring to DNA analysis for teeth. No? Oh, equipment is very expensive for teeth. I didn't know that. I'll do a bit of research on that to get more answers. For teeth, I saw that Leximon is collecting teeth in Leximon, so I don't know if they have that answer about where they send those. Would it be the department? We'd have to see about that. But I know they're doing this right now for moose. Well, that's really interesting. Leximon will be presenting tomorrow for moose. So you can ask that question tomorrow. We have a question from Rachel. It's just to answer about that teeth collecting. I work with Leximon, and my colleague Andréane will give a presentation on aerial surveys, and I've overseen tooth collection. It's not an inventory like we're seeing today, but we ask people to declare their catch, and we try to collect teeth through that. We haven't been successful yet, so this is a project that's just getting started. When we will have collected certain teeth, it's to send those to the department. Yes, we send those to the department right now. But of course, if we had all the equipment and the space required, we would do it ourselves. But nonetheless, it's something that requires a certain space and material for this. Right now, we're in partnership with the department for this. It was to answer that question. Thank you, Rachel. Okay, we'll continue. We'll have time to discuss at the end. So we're looking at camera trap survey. So all individuals are observed, or individuals aren't all observed. So we're looking at harvesting, and rather than use DNA for individuals, we'll look at physical characteristics of the individuals. So first of all, what is a camera trap survey? We'll install automatic cameras on the territory. So these cameras will be installed and detect movement. That's why we call them automatic. We leave them in the bush, and they'll take photographs when species pass in front. We'll often use those for hunting. So this will meet certain objectives, mainly to answer questions linked to species density or space use, the abundance of species. How is it done? That cameras are installed on the territory. So not a lot of field work to be done. And when do we carry out this survey? Mainly in the summer, but it's easier to take photos. But this can be done during all seasons. When we install a camera, we will ideally install it at the height of the species. If we study all of large fauna, we'll put it at a certain height so that we can capture wolves and moose, for example. For the moose, we'll see the bottom part of the body, and for the others, we'll see the whole body. The height will depend on the species or that you would like to capture by camera. Will be ideally a night mode without flash. We don't want a flash to disturb the individuals. So in infrared mode, this photo can be taken without modifying the behavior of the individuals. And The camera is triggered automatically with a movement. A picture is taken when an animal passes in front of the camera. The principle for this triggering is, yes, it will take a photo automatically but it will also take vegetation that moves in the wind. For example, here you have a camera that got triggered by all of this vegetation. So here we have photographs that, could just go on and on. And so you can have a photo of vegetation. So when you install a camera, it might be it's a good idea to cut out the vegetation in front of the camera. It would have avoided having 1,000 vegetation photos, because with cameras, once we have the photos at the office, we need to sort those. So if all we have is animals, the sorting will go a lot more quickly than if we have a whole bunch of photos and thousands of photos of vegetation that are part of the total number of photos. So why do we use cameras? We remember the method with the hairs, sampling hairs for individuals, but cameras, it's an alternative that's non-invasive compared to hair. We don't have to install traps that attract animals, and also it avoids laboratory analyses. So you don't have to send hair samples to labs to extract the DNA. You can use photos of individuals. That's the advantage. So with the hairs or fur for individual recognition, and with a camera, we'll use physical characteristics for individual recognition. The methods by camera were developed with tigers to start with. They would use this pattern of marking on the tiger to be able to capture those. When we are able to have individual physical characteristics, we can use cameras for inventories, and we'll use the same thing with the hair, with the fur. We'll put this into the software and we'll enter our individuals. If we have an example here with the black bear, the objective is to determine black bear abundance on a territory. So to recognize the individuals, we had markings at strategic areas at the level of their head to be able to recognize them. And then these markings identified in each of the photos that were taken were placed on the individuals. For the markings here, when we put them on each of the photos, we'll be able to say if these markings were at the same distance, it means it was the same individual. If they're not at the same distance, it means that it wasn't the same individual. So the marking is overlaid. And then we calculate the distances between these markings. And depending on these calculations, we're able to have an individual recognition. If all of the points here are always at the same distance for the same photo, it means it's the same individual. If they're not, it means it's another individual. After having identified the individuals, we enter these data in the software and we're able to see what the abundance is. However, with bears, individual recognition didn't really work. We were only able to recognize only half of the individuals, which is not very much. So we had to find another method to use cameras nonetheless, because this is what is less invasive. We don't want to get fur samples. Bringing cameras out on the field is nonetheless efficient and easy to do, and doesn't need a huge budget or a huge analysis. To do this, we tried to use cameras with no individual recognition. And how that works, we install the cameras and we'll have a number of photos of the species. For example, 20 photos of moose in the camera divided by the sampling time. So, for example, 100 cameras, 2 weeks in the bush, and it'll give us a photographic rate by day, by 20 days. It will give us a photographic rate for individuals. For example, in such a field, we'll have a photographic rate of 4 individuals per day for each of the cameras. With the camera, we'll have a detection surface, which allows us to calculate the individuals on that surface. And with that angle for photo taking and the distance at which the camera is able to take a photo, if they can take the photo of an animal up to 20 meters, it'll be 20 meters, and the angle is the angle that the camera can cover. This will allow us to calculate a detection surface. It's another part of sampling our surface area. And when we combine the photographic rate and the detection surface, and we add the traveling speed of individuals, we're able to determine a number of individuals by square kilometer. So it's an analysis method that's easy, not complicated, to determine the abundance of individuals. We can also do something else when we install cameras on the territory. We don't need to evaluate an abundance. We can just look at space use. Again, we'll use photographs. However, we can avoid calculating the density of individuals, and we can just look at photographic rate and compare that with habitat types to see how the species use the space. So here I'll use examples for this to be more simple. If we want to use this, we want to compare the use of forest roads by large fauna. This project was done where you want to see, here you see the types of forest roads. Here you have one that's less than 10 years and another one that's more than 40 years. So there are cameras that were installed along these forest roads to see how the use of forest roads changed over depending on the age. So there were photographic rates that were calculated for each cameras, and then a mean that was used for each of the roads or characteristics. And we see how the use of these roads changed depending on age. This was done for caribou, moose, gray wolves, black bears. And in these analyses, we looked at the difference between how old the roads were, what kind of tree, do they have deciduous trees? So depending on that environment, we were able to see what types of roads were more or less used by such and such a species. For example, the wolves would use more forest roads, which were newer. So there was not so much vegetation, for example. So that information was collected. Another example is to evaluate the efficiency of different restoration treatments. That was my master's degree, my master's thesis with the Isipit Council. We had 4 different treatments. The council closed some roads down and they allowed 4 different treatments, closed, decompacted, planted, and enriched ground soil, where they installed cameras in each of the roads. And then we calculated photographic rates for each of these roads, and we were able to compare between the different treatments to see which treatment was more efficient, which one was best for caribou, where we had a larger presence of caribou. And we were also able to see which treatment was impediment to the predators, such as the black bear and gray wolves. Other uses of cameras, it's a technology that is used more and more, not so invasive and not too expensive. Many things we could do with cameras. We can install cameras and at den entrances to determine exit date or the number of juveniles. It can be a species that uses that den each year. It helps us to identify the number of juveniles for each mother when the juveniles come out of the den. Also, we can install cameras at entrances of wildlife bridges or waterways to identify to what extent these bridges are used and waterways. So if we can evaluate the efficiency of these crossings for wildlife. Also, we can install cameras to collars. It doesn't have to be telemetric, but it can help us to determine individual diets with the camera. We see the caribou. Here's a photo. It's a camera on a cat. So you could see that the cat caught a mole here or a mouse. Cameras can also be installed to understand the behavior of certain individuals. That was done for caribou, for example. We can see the juveniles here, how much time they spend where and what they do, say, during the summertime, or how much time do they spend resting. It can help to evaluate behavior. Here, the order of magnitude of cost for cameras, DNA, and aerial surveys. So aerial surveys, it can be $9,500 or so to $15,000 per 100 square kilometers. For DNA samples, the cost of lab analysis it's at least $15 per sample and more. As mentioned earlier, it depends on the species and the number of samples. It can also depend on the question and size of the study area. Cameras, prices will vary generally between $125 and $225 each, $225, of course. The number of cameras depends on the question and also the size of the study area. You must also consider all the other material needed, the cost of field work, the human resources necessary, and the time required to produce and then analyze the data. So there we go. That's it for cameras. I'll just mention that at this time, we'll stop the recording